So we have talked about the chemical composition of the DNA, and now we look into the three-dimensional model of the DNA, which is called as the Watson and Crick model of the DNA. Let us see what does the three-dimensional structure look like. This is the structure of the DNA consisting of nitrogen bases, sugars, and phosphates, which are called as nucleotides. So the DNA is a long chain of polynucleotides, which we have analyzed so far. And these are two polynucleotide chains, which is the chemical composition of the DNA. If we look at the same structure three-dimensionally, then that is a model given by Watson and Crick, which is called as the DNA double helix model. That is a double helix model consisting of two threads which are parallel to each other and are spirally twisted around each other. So, two threads parallel to each other and spirally twisted around each other, which is called as the DNA double helix. First, let's see a little about the DNA. The DNA is a long polymer of deoxyribonucleotides, and you have bacteriophage called as 4 lambda 174 or 4174 having 5386 nucleotides. बैक्टीरियोफाज में बहुत कम न्यूक्लियोटाइड्स होते हैं. Bacteriophage lambda has 48,502 base pairs. So it is a little longer compared to 474. Then we have bacteria Escherichia coli, which has 4.6 into 10 raised to 6 base pairs. 4.6 into 10 raised to 6 base pair, and the haploid content of the human DNA is 3.3 into 10 raised to 9 base pairs. That is the amount of DNA present in one human DNA, haploid human DNA. By the way, a cell has in it two DNAs. One is maternal, one is paternal. So we have a total of 3.3 into 10 raised to 9 base pairs in the maternal DNA, 3.3 into 10 raised to 9 base pairs in the paternal DNA, giving you a total of 6.6 into 10 raised to 9 base pairs found in one cell, found in two DNAs of one cell. So that is the content and the number of base pairs. Let us discuss this DNA. In 1953, James Watson and Francis Crick proposed the DNA structure based on X-ray crystallographic studies provided by Morris Wilkins and Rosalind Franklin. So, two scientists, Morris Wilkins and Rosalind Franklin, did something called as the X-ray crystallographic study of the DNA. The X-ray crystallographic study was done, and using these studies, James Watson and Francis Crick proposed the DNA structure, which is called as the Watson and Crick model of the DNA, or the DNA double helix, which consists of two strands, as we just said, parallel to each other and are spirally twisted around each other, which is called as the DNA double helix. What does this DNA consist of? So here we have the chemical composition. This is the chemical composition. And this is the actual three-dimensional model of the DNA consisting of two DNA strands which are spirally twisted And that spiral twisting, we remember, is called the double helix. What does the DNA show? The DNA molecule as a double helix, according to the model proposed by Watson and Crick, one DNA molecule consists of two long strands coiled around a imaginary central axis to form a double helix, which looks like a twisted ladder. तो ये है एक इमेजिनरी सेंट्रल एक्सिस और यहां है दो डीएनए स्ट्रैंड. The two DNA strands are in a right-handed coiling and it coils, 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 coils like a double helix. तो ये आपका डबल हेलिक्स मॉडल बन जाता है. So this is the imaginary coiling around the central axis. And that is the three-dimensional structure of the DNA. What is the structure of the DNA made up of? Well, that we know everything. The strand consists of a number of nucleotides, to be specific, deoxyribonucleotides. Each nucleotide is made up of a deoxyribose sugar, phosphate group, and a nitrogen base. It's made of two polynucleotide strands. The backbone is made up of sugar phosphate and the bases project inside. What is the strand made up of? The strand is made up of sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, and the nitrogen bases are present inside. So the backbone is made up of sugars and phosphates. And what is the continuation? The successive nucleotides of the same strand are linked together by three phosphodiester bonds. And phosphodiester bonds से दो nucleotides attached होते हैं. Phosphate attached to the fifth carbon of one nucleotide is joined to the third carbon of the next nucleotide by phosphodiester bonds. Then we have complementary base pairing. The nitrogen bases present on one strand always pair with nitrogen bases of the opposite strand. जिसके अंदर purine adenine always pairs with base pair pyrimidine thymine by two hydrogen bonds, and the purine guanine always pairs with the pyrimidine base cytosine by three hydrogen bonds. So A pairs with T by doublet, two hydrogen bond. G pairs with C, triplet, three hydrogen bond. Double ring purine pairs with single ring pyrimidine thymine. Adenine, double ring purine pairs with single ring pyrimidine thymine, and double ring guanine pairs with single ring cytosine. A और T के बीच में टू हाइड्रोजन बॉन्ड, G और C के बीच में थ्री हाइड्रोजन बॉन्ड्स. So you have doublets and triplets. Between AT, doublets, and between GC, triplets. And that is called as complementary base pairing. So if you see the DNA, entire DNA has got same pairing. A pairs with T, A pairs with T, T pairs with A, G pairs with C, C pairs with G. तो सर वो भी स्पेसिफिक है. G pairs with C by three hydrogen bonds, C pairs with G by three hydrogen bonds, A pairs with T by two hydrogen bonds, T pairs with A by two hydrogen bonds, and always a purine pairs with a pyrimidine. So that is always golden. अगर एक स्ट्रैंड पे प्यूरीन है, ऑपोजिट स्ट्रैंड पे pyrimidine होता ही है. अगर यहाँ प्यूरीन है तो यहाँ pyrimidine है. अगर यहाँ pyrimidine है तो यहाँ प्यूरीन है. And a purine always pairs with pyrimidine. That rule is golden. So that is called as complementary base pairing. It's called as complementary base pairing. And the two strands are complementary to each other. Two strands are complementary to each other. Why? Suppose I know there is a thymine here, there has to be adenine here. If there is guanine here, there has to be cytosine here. If there is adenine here, there has to be thymine here. If there is guanine here, there has to be cytosine here. If there is cytosine here, there has to be guanine here. If there is adenine here, there has to be thymine here. If there is thymine here, there has to be adenine here. तो अगर एक सीक्वेंस पता है, if you know the sequence of one strand, you automatically know the sequence of the other strand. So both strands are exactly matching to each other. They are matching because of complementary base pairing. As they are matching, they are called as complementary to each other. The two strands are complementary to each other. The two strands are complementary to each other. And then, sir, you have complementary base pairing. Then we have the logic. The nitrogen bases along with the hydrogen bonds make the rungs of the ladder. Backbone कौन है? Sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. They form the backbone. And उनके बीच में nitrogen bases form the rungs of the ladder. तो ये है nitrogen bases. They form the rungs of the ladder with hydrogen bonds in between. तो सर पिक्चराइज करने के लिए सपोज वी हैव एक ओल्डन जमाने का स्टेयरकेस. तो वुडन स्टेयरकेस जो होता है, एंड आप वुडन स्टेयरकेस के दोनों साइड बार्स को पकड़ते हो एंड यू पुट योर स्टेप. आप उसके ऊपर एक वुडन बार के ऊपर स्टेप डालते हो. यू पुट वन स्टेप ऑन द वुडन फ्लोरिंग एंड यहां दो फ्लावर पॉट है, यहां एक फ्लावर पॉट है. नेक्स्ट स्टेप लिया तो वुडन स्टेप के ऊपर यहां एक फ्लावर पॉट है, यहां दो फ्लावर पॉट है. नेक्स्ट स्टेप लिया, यहां एक फ्लावर पॉट है, यहां दो फ्लावर पॉट है. नेक्स्ट स्टेप लिया, यहां दो फ्लावर पॉट है, यहां एक फ्लावर पॉट है. ऐसे कंटिन्यूसली आप ग्राउंड फ्लोर से फर्स्ट फ्लोर जाओ, फर्स्ट फ्लोर से सेकंड फ्लोर जाओ, आपको 10 वुडन स्टेप्स मिलेंगे. सो 10 वुडन स्टेप्स से आप ग्राउंड फ्लोर से फर्स्ट फ्लोर जाते हो. अनदर 10 वुडन स्टेप्स से आप फर्स्ट फ्लोर से सेकंड फ्लोर जाते हो. अनदर 10 वुडन स्टेप्स से आप सेकंड फ्लोर से थर्ड फ्लोर जाते हो. तो वो जो आपने साइड में रेलिंग्स पकड़ के रखी है, व्हाट आर दोज रेलिंग्स जो आपने पकड़ के रखी है? alternately arranged sugar and phosphate. Alternately arranged sugar phosphate, they form the backbone. वो फ्लावर पॉट्स कौन है वुडन स्टेप्स के ऊपर? नाइट्रोजन बेसेस. नाइट्रोजन बेसेस आर द फ्लावर पॉट. जहां दो फ्लावर पॉट है प्यूरिन, जहां एक फ्लावर पॉट है पिरिमिडीन. जहां एक फ्लावर पॉट है पिरिमिडीन, जहां दो फ्लावर पॉट है प्यूरिन. एंड उनके बीच में वुडन स्टेप्स है. वुडन स्टेप्स आर द हाइड्रोजन बॉन्ड्स इन बिटवीन. सो दैट ब्यूटिफुल स्टेयरकेस इज योर डीएनए. और एक टर्न के अंदर कितने बेस पेयर्स होते हैं? यू हैव वन टर्न हैविंग इन इट, ध्यान से सुनना ऑल ऑफ यू. This is one turn having in it a total of, check it here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 base pairs are present in one turn. तो एक टर्न के अंदर there are 10 base pairs. In one turn, there are 10 base pairs. 10 base pairs are present in one turn. तो ये देखना वापस 1, ये छुप गया है सेकंड, थर्ड, फोर्थ, फिफ्थ, सिक्स्थ, सेवेंथ, एट, नाइन्थ एंड टेंथ. ऐसे एक टर्न के अंदर you are having a total of 10 base pairs. In one turn, you are having a total of 10 base pairs. And that is your three-dimensional structure of the DNA. ऐसे एक 3D structure होता है. And sir, next we have the purine pyrimidine ratio. Due to complementary base pairing, the total number of purines is always equal to the total number of pyrimidines, which is a 1:1 ratio, which is called as Chargaff's rule. इसे Chargaff's rule कहते हैं. A plus G equal to T plus C. The ratio of purine equal to the ratio of pyrimidine. Or in other words, A plus G upon T plus C is always 1. Purine upon pyrimidine ratio is always 1. That is called Chargaff's rule. Why, sir? Now, if you check it here, if you have a purine here, if you have purine here, you always have pyrimidine here. If you have pyrimidine here, you always have purine here. If you have purine here, you always have pyrimidine here. So if you count all the number of purines, 1, 2, 3, 4, 5, 6, 7. So how many A plus G are there? 7. Count it to the number of pyrimidine T plus C. 1, 2, 3, 4, 5, 6, 7. So how many T plus C are there? T plus C are 7. So A plus G equal to T plus C, or in other words, A plus G upon T plus C is 7 upon 7, always equal to 1. And this rule is called as the Chargaff's rule. Chargaff's rule that the purine pyrimidine ratio is constant and always equal to 1. And check this carefully here. That's called Chargaff's rule. So that is Chargaff's rule. Then we have polarity of each strand. Each strand of the DNA has a specific polarity. One end is called as C5 end, whereas the other end is called C3 end. The polarity is because of third carbon and fifth carbon. At fifth carbon, there is a free phosphate group. And at the third carbon, there is a free hydroxyl group. So check the polarities of the two strands. अगर polarity देखी जाए, check this carefully. Sir, ये है ध्यान से oxygen, first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and you have nucleotide, nucleotide, nucleotide. ऐसे polynucleotides है. Every phosphate is attached to two sugars. Fifth carbon at one nucleotide, third carbon of next. So phosphodiester, 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 phosphodiester. But after covering 3 into 0.3, 3.3 into 10 raised to 9 base pair, when we come to the uppermost nucleotide, uppermost nucleotide हम लोग जब पहुंचते हैं, this is the uppermost nucleotide attached to fifth carbon. Fifth carbon. Now इसके ऊपर और एक न्यूक्लियोटाइड नहीं है, तो यहाँ phosphodiester bond नहीं बनता है. There is no phosphodiester bond. Rather, this is a phosphoester bond. So every bond has a phosphodiester bond. Every bond is a phosphodiester bond. But at one end, there is not a diester bond, there is a phosphoester bond. Phosphoester bond. Everywhere else, it was phosphodiester. सब phosphates में phosphodiester था, but this is phosphoester bond. तो ये phosphate थर्ड कार्बन पे अटैच नहीं है because this is the uppermost nucleotide. So at the 0, C1, C2, C3, C4, C5, fifth carbon पे there is a free phosphate. Fifth carbon पे there is a free phosphate. At fifth carbon there is a free phosphate, and this free phosphate is called as the free phosphate end. At free phosphate end, which carbon is that phosphate attached to the fifth carbon? That phosphate is attached to the fifth carbon. Hence, this end is called as the C5 end. So, C5 end has a free phosphate group. C5 end has a free phosphate group. And check this here. At every third carbon, at every third carbon, what was present? At every third carbon, there was a hydroxyl present. Third carbon पे there is a hydroxyl present. But that hydroxyl, ये है hydroxyl. That hydroxyl was used for condensation to form a water molecule and get attached to the phosphate to form a phosphodiester bond with the fifth carbon of the next sugar. तो थर्ड कार्बन पे जो हाइड्रोक्सिल था, इट कंडेंस विथ OH and H to form water. तो जितने भी थर्ड कार्बन है उसका हाइड्रोक्सिल यूज हो जाता है, थर्ड कार्बन का हाइड्रोक्सिल यूज हो जाता है टू फॉर्म वाटर एंड फॉर्म अ फॉस्फोडाइस्ड बॉन्ड. तो everywhere we are having third carbon OH is used for phosphodiester bond. Third carbon phosphate ka OH for the phosphodiester bond. OH used for 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 phosphodiester bond. And water molecules are formed. But this is the last nucleotide. Nitrogen, sugar, and phosphate. This phosphate is attached to the fifth carbon of the nucleotide and to the third carbon of next nucleotide. But इसके third carbon पे there is a hydroxyl और ये लोअर मोस्ट न्यूक्लियोटाइड है, तो इसके नीचे नया फॉस्फेट शुगर और नाइट्रोजन बेस नहीं है. सो दिस OH does not get condensed to form a water molecule, क्योंकि ये तो लास्ट हाइड्रोक्सिल है एंड लास्ट न्यूक्लियोटाइड है. तो यहां हाइड्रोक्सिल यूटिलाइज नहीं होता है to form a phosphodiester bond. So this is a free hydroxyl. Everywhere else, hydroxyls were used for condensation to form water molecule, but here we have a free hydroxyl. At which carbon is that free hydroxyl present? At the third carbon. So that end is called as the C3 end. C3 end. So what do we have at the C3 end? A free hydroxyl group. At C3 end, we have a free hydroxyl group. And at the C5 end, we have a free phosphate group. C5 end, we have a free phosphate group with a phosphomonoester bond instead of phosphodiester bond. And phosphomonoester bond instead of phosphodiester bond. And the third carbon, sorry, we have a free hydroxyl group, and that will be called as the C3 end. A free phosphate at the C5 end, a free hydroxyl at the C3 end. So these are the two ends. Now check this carefully. In one of the strands, oxygen is facing upward. So rule of thumb, the first carbon which comes in the clockwise direction is the C1. So if this is oxygen, this is C1, this is C2, this is C3, this is C4, this is C5. Oxygen here, one, second carbon, third carbon, fourth carbon, fifth carbon. And if this is oxygen, first, second, third, fourth, fifth carbon, where is the C5 end present? Above. And this is oxygen, C1, C2, C3. And where is the free hydroxyl present? Below. So, ये है C3 end to C5 end, C3 to C5 going upwards. C3 to C5 going upward is called as the ascending strand. ये strand कौन सा हो जाता है? Ascending strand. And 3 to 5 is always the polarity. So always polarity is 3 to 5 direction. So C3 किधर है? नीचे. और C5 किधर है? ऊपर. So what is that strand called as? The ascending strand. But if we look at the opposite strand, यहाँ ये strand में oxygen ऊपर था, but in the opposite strand, oxygen is facing downwards. Oxygens are facing downward. So rule of thumb, in the clockwise direction, if this is oxygen, clockwise, this becomes C1. This becomes C2. This becomes C3. This becomes C4. This becomes C5. And first nitrogen base is attached to carbon number 1 by glycosidic bond. But where is the free OH present at the C3 end? Above. So C3 end ऊपर होता है, and you have oxygen, carbon 1, 2, 3, 4, 5. Where is the free phosphate present? The free phosphate everywhere phosphodiester. Phosphodiester, 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 phosphodiester. But यहाँ phosphoester, phosphoester. So what is this end called as? C5 end. So here we have O, C1, C2, C3, C4, C5, and here we have C5 end. So O, 1, 2, 3, 4, and fifth carbon पे free phosphate होता है. So this is the C5 end, and यहाँ O, 1, 2, 3, 4, 5. At the third end, there is a free hydroxyl present. Third end pe there is a free hydroxyl present. So this is the C3 end. So ये strand में oxygens ऊपर थे. Because oxygens were present above, we had 1, 2, 3 and OH present below. And we had 1, 2, 3, 4, 5 phosphate present above. So it was C3 below and C5 above, and that was called as ascending strand. ये ascending strand था. But in the opposite strand, as the oxygens are facing below, you have C1, C2, C3, C4, C5, and free OH present at third carbon above. So C3 ka end is above, and we have O, C1, C2, C3, C4, C5, and phosphate present below. So C5 end is below. So इस strand में OH यानी C3 end is above, and O 1, 2, 3, 4, 5 phosphate is present below. As phosphate is present below, that becomes the C5 end. So where is the polarity now? C3 to C5 is in the downward direction. C3 to C5 is in the downward direction. Hence, this strand is called as the descending strand. So, if you remember, here we have OH down and C3 end. And here we have C5 up, OH down, and C3 end. And here we have C5 up, attached to it free phosphate, and that strand is called as the ascending strand because 3 to 5 polarity is above. Yaha we have oxygen facing upwards. Oxygen facing upward. Yaha oxygens are facing downward. So we have OH present here. So ye has C3 end. And oxygen present here. So here we have phosphate present here. 1, 2, 3, 4. Phosphate present here. We have C5 end. And 3 to 5 downward. What is that strand called as? The descending strand. So the two DNA strands are parallel to each other, but they are parallel in the opposite natures. They are parallel in the opposite nature. One strand is ascending and one strand is descending. One strand is ascending, the other strand is descending. So the two strands are called as anti-parallel in nature. The two strands are anti-parallel in nature. So they are ascending, descending, they are anti-parallel in nature. तो ये ध्यान से देखना सर, ये है आपका C3. So which is this strand, C3 to C5 downward. So ये arrow marks भी दिखाया हमने. Arrow down, 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 down. So C3 to C5 downward. What is this strand called as? The descending strand. And parallel to it, but in the opposite direction. Here we have C3 and here we have ascending, going towards C5. And this strand is ascending upwards and is ascending upward. So this is C3 to C5 upward, and that is called as the ascending strand. So both the strands are parallel but in the opposite direction. Hence they are called as anti-parallel in nature. The two strands are called as anti-parallel in nature. So that is anti-parallel nature. Then we have grooves, major and minor grooves. The DNA strands are a right-handed coiling around a central imaginary axis. So, ये है imaginary axis, ये है मेरा right hand, और ये ऐसे राइट हैंडेड कॉइलिंग होता है अराउंड द इमेजिनरी सेंट्रल एक्सिस. And the coiling of the double helix results in the formation of major or deep grooves. And the twisting of strands around each other forms minor or shallow grooves which are found in an alternate manner. तो ध्यान से सुनना ये all of you. This is difficult to make you visualize. So space between the two DNA strands, space between the two DNA strands is called as the minor groove. तो ये है माइनर ग्रूव. यहां देखना ध्यान से. ये है माइनर ग्रूव, माइनर ग्रूव, माइनर ग्रूव, माइनर ग्रूव, माइनर ग्रूव, माइनर ग्रूव, माइनर ग्रूव. तो space between two strands is called as the minor groove. दोनों strands के बीच की space है. This space is called minor groove. 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 So space between two strands is called as minor groove. Space between two strands is called as minor groove. तो ये कंटिन्यूसली हमने क्या दिखाया है? माइनर ग्रूव. एंड माइनर ग्रूव इज about 12 angstrom units. 12 angstroms. You've done it in physics. How much is 1 angstrom? 10 raised to minus 10 meters is 1 angstrom. तो कैन यू इमेजिन हम लोग कितने मॉलिक्यूलर लेवल पे, क्या माइक्रोस्कोपिक लेवल पे बात कर रहे हैं. दिस इज 12 एंगस्ट्रॉम यूनिट्स, सो स्मॉल, एंड दैट इज द माइनर ग्रूव. सो माइनर ग्रूव इज 12 एंगस्ट्रॉम्स एंड दैट इज द स्पेस बिटवीन टू डीएनए स्ट्रैंड्स. एंड नाउ ध्यान से यहां से लेके यहां, check this here. This is one double coil. This is one double coil. And this space of a double coil is called as a major groove. Once again, check the fingers. Check the finger. Lower fingers here gone. This space is called as major groove. This space is called major groove. So space in one coil is called as major groove. Space in one coil of the double helix is called the major groove. Space in one coil of double helix is called the major groove. So once again, what is space between two strands called as minor groove? So what is the space between two strands called as minor groove? Space between two strands is called minor groove. And space in one double helix, space in one double helix, space in one double helix is called major groove. So if you see diagrammatically, space in between two strands is called minor groove, and this is space of one double helix. तो ये डबल हेलिक्स इधर था, डबल हेलिक्स इधर है, डबल हेलिक्स इधर है, और दूसरा डबल हेलिक्स इधर है, डबल हेलिक्स इधर है. सो एक कंप्लीट टर्न हुआ है, and that space is called as a major groove. So space between two strands is called as minor groove. Space between two strands is called minor groove. And space between a double helix, space between a double helix is called as a major groove. Minor groove is 12 angstrom units. Major groove is 22 angstrom units. ये notes में लिख लेना, not given in the notes. Minor groove is 12 angstrom units. Major groove is 22 angstrom units. So if you now see this, so ये कौन सा space है? Minor groove, which is 12 angstroms. और ये कौन सा space है? Major groove, which is 22 angstroms. So now check this here. This is one spiral of one strand. ये चेक करना. ये कौन सा ग्रूव है? माइनर. ये कौन सा ग्रूव है? माइनर. और ये कौन सा ग्रूव है? मेजर. मेजर, माइनर, माइनर, मेजर. अब अगर हम लोग डबल स्पाइरल की जगह सिंगल स्पाइरल ले लें, सो चेक माय फिंगर. दिस इज मेजर ग्रूव, मेजर ग्रूव, मेजर. But now see the fingers I'm removing. Now see the finger. Lower finger here, lower finger here. This turn. This is one spiral of one strand. One spiral of one strand. This is one spiral of one strand. This was minor. This was minor. This was major. So one spiral of two strands was a major groove. One spiral of two strands was a major groove. But now I'm referring to one spiral of one strand. One spiral of one strand. How much is that? Check it here. ये था minor, minor, major. And one spiral of one strand is a minor groove plus a major groove. One spiral of one strand is a minor groove plus a major groove. So check this here. ये है minor, ये है major, and one minor, one major, one spiral of one strand. One spiral of one strand is minor plus major. And how much will that be? If you remember, minor was 12 angstrom units, major was 22 angstrom units. So one spiral of one strand will be 12 plus 22, a total of 34 angstrom units. And one spiral of one strand is 34 angstrom units, which is called as the pitch of the DNA. चलो ऐसे जरा देखते हैं. Yeah, so we had one minor plus one major. How much is minor groove? 12 angstrom units. How much is major groove? 22 angstrom units. So how much is one spiral of one strand? Check this here. This is minor groove, 12 angstroms. This is major groove, 22 angstroms. And this is minor is between two strands. Major is in between one double spiral ka space. And now you have minor plus major. Minor plus major is one spiral of one strand. This is one spiral of one strand. One spiral of one strand includes minor groove and major groove. This is distance, minor groove. This is major groove. So how much will that be? It will be 12 angstroms plus 22 angstroms. A total of 34 angstroms will be the distance of one spiral, and that is called as the pitch of the DNA. So how much is the pitch of the DNA? एक स्पाइरल का लेंथ, एक स्पाइरल का लेंथ, वन स्पाइरल ऑफ वन स्ट्रैंड, वन स्पाइरल ऑफ वन स्ट्रैंड, वन स्पाइरल ऑफ वन स्ट्रैंड, वन स्पाइरल ऑफ वन स्ट्रैंड. इसके ऊपर दूसरा स्पाइरल है, इसके नीचे दूसरा स्पाइरल है. So this is another spiral, this is another spiral, and this is one spiral of one strand. One spiral of one strand. One spiral of one strand, which is called pitch of the DNA. And pitch of the DNA is minor groove plus major groove, 34 angstrom units. 34 angstrom units. So this is the pitch of the DNA, which is minor groove plus major groove, a total of 34 angstrom units. And 34 angstrom units may how many nucleotides are present? How many nucleotides are present? That depends on how many hydrogen bonds we see in the between. तो याद है हमने एक स्टेयरकेस जब चढ़ा था तो हमने ग्राउंड फ्लोर से फर्स्ट फ्लोर कितने स्टेप्स लिए थे? 10 स्टेप्स. फिर फर्स्ट फ्लोर से सेकंड फ्लोर कितने स्टेप्स लिए थे? 10 स्टेप्स. सेकंड फ्लोर से थर्ड फ्लोर कितने स्टेप्स लिए थे? 10 steps. That is exactly the same. तो एक पिच के अंदर कितने न्यूक्लियोटाइड्स है? Check it here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So a total of 10 nucleotide pairs are present in 34 angstrom units. So a total of 10 nucleotide pairs. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. So a total of 10 nucleotide pairs are present in 34 angstrom units. So total of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 nucleotide pairs are present in 34 angstrom units. तो एक न्यूक्लियोटाइड कितना होगा? एक न्यूक्लियोटाइड कितना होगा? 34 divided by 10 means a total of 3.5 3.4 angstrom units is the dimension of one nucleotide. One nucleotide is 3.4 angstrom units. One nucleotide is 3.4 angstrom units. This is 3.4 angstrom units, which is the amount of DNA or the structure found in one nucleotide. तो एक न्यूक्लियोटाइड में कितना होगा 3.4 angstrom units. And check it here, the DNA molecule के अंदर one nucleotide is having a distance of how much? 3.4 nanometer. That is the entire DNA. We'll just come to that. And first, okay, nanometer conversion बोलता हूँ. So one spiral DNA has in it how many nucleotides? One DNA, one spiral of one DNA is 34 angstrom units and has in it 10 pairs of nucleotides. So one nucleotide is 3.4 angstrom units. 3.4 angstrom units. ये याद रखना. I hope you have understood this point. So what is space between two strands called? Minor groove. What is space between two strands called? Minor groove. What is distance of one double spiral called? Major groove. And how much is a minor groove plus a major groove called as the pitch of the DNA? Minor plus major is called as pitch of the DNA. How many angstrom unit it is? 34 angstrom. How many nucleotides in it? 10 nucleotides. So how much is one nucleotide? One nucleotide is 3.4 angstrom units. This was most crucial today. And check this here, the diameter. The diameter of the DNA is 20 angstrom units or 2 nanometers. The diameter of DNA is 2 nanometers. 1 nanometer is 10 raised to minus 9 meter. And 1 angstrom unit is 10 raised to minus 10 meters. So 1 nanometer is 10 angstrom units. 1 nanometer is 10 angstrom units. ये भी याद रखना. So DNA का diameter कितना है? 2 nanometer. DNA का diameter है 2 nanometer. In other words, 20 angstrom units. The diameter of the DNA is 2 nanometers, or in other words, 20 angstrom units is the diameter of the DNA. Diameter of DNA is 2 nanometers or 20 angstrom units. And how much is the angle between two nucleotides? So, sir, what is the angle? Spiral ladder of the DNA is due to deflection angle of 36 degree between two successive steps. Sir, if you remember, एक complete टर्न के अंदर कितने न्यूक्लियोटाइड पेयर्स हैं? हमने ग्राउंड फ्लोर से फर्स्ट फ्लोर कितने वुडन स्टेप्स चढ़े थे? ग्राउंड फ्लोर से फर्स्ट फ्लोर फ्लावर पॉट वाले कितने वुडन स्टेप्स चढ़े हमने? 10. तो 10 wooden steps form one complete cycle or turn. And what is the angle of one turn? 360 degrees. और उसके अंदर कितने स्टेप्स है ऐसे, और कितने न्यूक्लियोटाइड पेयर्स है डीएनए में? 10 pairs. So what is the angle between two successive pair? 36 degrees. So 36 degree, 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 ऐसे total 36 degrees into 10, become one spiral. So the angle between two nucleotide pairs, angle between two nucleotide pairs is 36 degrees. And they cover a distance of 3.4 nanometers, or in other words, 34 angstrom units. So that we did already. कितने एंगस्ट्रॉम यूनिट्स है? 34 एंगस्ट्रॉम. उसको नैनोमीटर में दिया है एनसीईआरटी में. सो हाउ मच इज इट? 3.4 nanometer. 3.4 nanometer. And one nucleotide will be how much? One nucleotide is 3.4 angstrom units. In other words, 0.34 nanometer. तो ये सिर्फ कन्वर्जन याद रखना. हाउ मच इज द पिच ऑफ द डीएनए? 34 angstrom units, which is 3.4 nanometer. 10 raised to minus 10, 10 raised to minus 9 nanometer angstrom. So 34 angstrom units is 3.4 nanometer is the pitch of the DNA. Pitch of DNA. And the distance of one nucleotide or dimension of one nucleotide, dimension of one nucleotide how much is dimension of one nucleotide? 0.34 nanometer or 3.4 angstrom units. 3.4 angstrom unit or 0.34 nanometer. एक पिच के अंदर कितने हैं? एक पिच ऑफ डीएनए में न्यूक्लियोटाइड्स कितने हैं? देयर आर 10 pairs of nucleotides. 10 pairs of nucleotide. So if 34 angstrom units is 10 pairs, then one nucleotide is 0.34 nanometer or 3.4 angstrom unit. 0.34 nanometer or 3.4 angstrom units. And this is the entire logic of the DNA. DNA हमने इतना डिटेल में पढ़ा है, we have understood this completely. We end this with a beautiful logic. Segments of DNA are called as genes. We always knew this. But now we know everything about DNA. DNA is nothing but nucleotide pairs. Pairs of nucleotides. DNA is nothing but nucleotide pairs connected by hydrogen bonds. So, a gene is nothing but some number of nucleotide pairs. Some number of nucleotide pairs form one gene. How many nucleotide pairs? So if you remember, total एक डीएनए में कितने न्यूक्लियोटाइड्स है? ऐसे एक डीएनए हैज 3.3 into 10 raised to 9 base pairs. 3.3 into 10 raised to 9, 3.3 billion base pairs are present in one DNA. इतना लंबा होता है आपका डीएनए मॉलिक्यूल, उसमें 3.3 into 10 raised to 9 base pairs है. तो हमने ये जो दिखाया है is only 7 base pairs. There are a total of 3.3 into 10 raised to 9, 3.3 billion base pairs. इतना लंबा डीएनए है. हमने तो सिर्फ एक माइक्रोस्कोपिक पार्ट दिखाया आपको. And how much is a gene? First base pair, second base pair, third base pair, fourth, fifth, sixth, seventh, 8, 9, 10, 11, 12, 13, 14, 15, approximately 3 1000 base pairs form one gene. 3000 base pairs form one gene. 3000 base pairs का एक जीन होता है, and we have around 30,000 such genes in the entire human DNA. 30,000 genes in the entire DNA. Each gene is 3,000 base pairs. And अलग अलग जीन से अलग अलग बॉडी पार्ट्स बनते हैं. Different different genes from different body parts. अरे but sir, how do these genes differ from each other? Sugar is same, phosphate is same, nitrogen bases are same, adenine, guanine, thymine, cytosine, sugar, deoxyribose sugar, phosphate, phosphoric acid. So how does one gene differ from another gene? All the genes are same. Suppose you have the gene for color of eye. Gene for color of eye found on chromosome number 1. It consists of approximately 3000 nucleotide pairs, and समझो the sequence of nitrogen bases is TA, GC, AT, AT, CG, GC, TA, AT, AT, GC, GC, TA, 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 GC, 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 TA, 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 GC, GC, AT, 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 TA, TA, and so on. And that gene is for color of eye. It say forms black color eye, black color. एंड समझो उसके अंदर टीए, जीसी और एटी की जगह इट बिकम्स टीए, टीए. Then now what is the sequence? Instead of TA, GC, AT, AT, CG, GC, TA, it has now become TA, GC. Instead of AT, it has become TA. Instead of AT, it has become TA. And then it forms CG, GC, TA, etc. And the gene has undergone a variation. And instead of black color, that TA will produce a blue colored eye. Instead of black color, एक blue color eye baby हो जाता है. Can you imagine? सिर्फ एक AT has undergone a change to form TA, and black has become blue. And that means one gene differs from another gene depending only on the sequence of the nucleotide. Nucleotide sequences अलग अलग होते हैं, तो ये बेस पेयरिंग्स अलग होते हैं. समझो you have gene number 1, as we said, is for color of eye. Gene number 30,000, total 30,000. So gene number 15,283 is say responsible for forming keratin. तो केराटिन के अंदर सीक्वेंस क्या है? केराटिन में सीक्वेंस है TA, GC, and got GC, CG, TA, AT, TA, which is different from the gene for color of eyes. And keratin का gene का sequence अलग हो गया, and that sequence forms keratin. Some other sequence, say in the muscles, the sequence is TA and again TA and again GC and again AT. चलो वो सेम रखते हैं, and then TA and then CG and then AT. So this sequence has also become different, and this sequence forms actin and myosin. There we have understood sugars are same, phosphates are same, but the only difference between genes is sequence of nucleotides. नाइट्रोजन बेस पेयर्स का जो सीक्वेंस है वो एक दूसरे से अलग होता है और उससे अलग अलग जीन्स बनते हैं and different genes form different characteristics of the body. In other words, four magical alphabet: thymine, adenine, guanine, cytosine, ATGC, इनके अलग अलग परम्यूटेशन कॉम्बिनेशन से अलग अलग जीन्स बनते हैं एंड अलग अलग जीन से हमारे बॉडी के अलग अलग कैरेक्टरिस्टिक्स बनते हैं एंड वी all are having different characteristic only based on a permutation and combination of four alphabets ATGC. Wow! But how? Wow! But how? How can just four alphabets make such a difference? और एक जाइगोट से अलग अलग जीन्स विच आर शुगर सेम फॉस्फेट सेम, हाँ ओनली उन जीन्स के अंदर सीक्वेंस ऑफ ATGCs were different और ATGC sequences were different एंड उनसे अलग अलग जीन से बॉडी के अलग अलग पार्ट्स बने and they were created as a perfect human baby from one cell called zygote with so many proteins all different from each other and all proteins nothing but a permutation combination of amino acids and that was decided by sequence of DNA having four alphabets ATGC. Wow! But how? इस चैप्टर में around 3, 4 say 5 lectures later you will understand how four alphabets form different types of proteins and make our body from one cell called the zygote. Absolutely fantastic concept. The secret of life itself, the beauty of life lies in the sequence of four alphabets ATGC. How tall you are, how good looking you are, what is the shape of your nose, what is the color of your eyes, how much is your intelligence, everything, everything depends on the permutation and combination of four alphabets ATGC, and that wow is something we are going to understand as how in a few lectures from now. Chalo. So today what we have done is very simple. ये तो इतना easy था. ये सब स्कूल में किया है, बायोमोलिक्यूल में किया है. Next session say we will step it up where we start understanding how adenine, guanine, thymine, and cytosine form different types of genes and understand the logic of protein synthesis in detail. Chalo. I hope you have understood this well. See you all soon. Take care.